Okay, this is Module 7, and it's Hematology. Hematology is the study of the body's blood cells, and there's three types that you need to know. Of course, there's more different cells in the body, but these are just found in the blood. The erythrocytes are, that's another name for red blood cells, and this is what they look like on a stained smear. They are responsible for carrying oxygen, and if the number of red cells is decreased, the patient is anemic, so they're said to have anemia. The leukocytes are the white blood cells, and these are two in the center. Um, if the white cells are decreased, that term is called leukopenia. If they're increased, it's called leukocytosis. And if you notice, if anytime you see penia at the end of a word, that means a decrease, and cytosis means an increase. Leukemia is when a patient has a malignant increase in leukocytes, so they have an abnormal amount of white cells in their bloodstream. And then the thrombocytes are another word for platelets, and these are these really small spots right here. An increase in those is called thrombocytosis, and a decrease is thrombocytopenia. So the red cells carry oxygen, the white cells fight off infection, and the thrombocytes are involved in clotting. We count red cells and white cells, not platelets. We count these cells in blood, but also other body fluids too. We do spinal fluid cell counts. The spinal fluid comes from the spinal column. That's the fluid that surrounds the brain in the nervous system. We do counts on serous fluids, which are the fluids that are around the heart, around the abdominal cavity, and around the lungs. And those are pericardial, peritoneal, and pleural. We do synovial fluid counts, which is the joint fluid. Most of the time we do knees, but we can do wrist or elbows or any joints. And we also do, which is really considered a tissue, but we do test on bone marrow. This is considered myelogenous tissue, and the bone marrow, of course, is found inside the bone, and this is where the white cells, red cells, and platelets are formed. This is the only tissue that we do counts on. When the physician suspects something's going on with the patient's red cells or white cells or platelets, they can get a bone marrow specimen, and we can put it on a slide, and we can look at it, and we can do a count, and it's usually evaluated by the pathologist. Okay, in the blood cells, the average amount of blood in an adult is 6 liters. 45% of that is red cells, white cells, and platelets, and 55% is plasma. You need to know this term, hematopoiesis. Hematopoiesis is the making of the cells. So poiesis means to generate or to produce. So hematopoiesis is the production of the cells. And this is done, like I said, in the bone marrow. In the bone marrow are where we found pluripotential stem cells. The stem cells are like the very first early cells, and they have the potential of becoming any type of cell that they're programmed to become. All of the blood cells mature in the bone marrow except for lymphs. So the red cells and the other white cells and the platelets are created in the bone marrow, and they stay in the bone marrow until they're mature and it's time for them to be released into the bloodstream. And then they're released into the bloodstream and they do their job. The lymphocytes are produced in the bone marrow, but they leave and they mature in the lymph tissue, and then they're released after that. Okay, erythrocytes or red cells, of course, they carry oxygen. They carry oxygen to all the tissues in the entire body. They have a nucleus until the last two stages, so a mature red cell does not have a nucleus. The average lifespan is 120 days. After the 120 days, the red cells start to break down and they're removed from the bloodstream by the spleen. The spleen, the red cells are broken down into several different components, and most of those are reused by the body, except for bilirubin. Bilirubin is not reused, and it has to be eliminated. So bilirubin is taken to the liver. The liver does some stuff to it, and bilirubin is normally released in the intestines in your stool. In cases of abnormal problems, the bilirubin can be found in the urine and it goes out through the kidneys. But again, that's not normal. That when bilirubin is found in the urine, something's going on with the liver or the red cells or something through that whole process. The leukocytes, like I said, they fight infection. They fight off anything foreign that gets into your body. And red cells last 120 days. White cells only last 10 or less hours, which is not a lot. There's different types of white cells that you need to know. The main three are monocytes, granulocytes, and lymphocytes. And then the granulocytes, there are three different types, and these are the neutrophils, the basos, and the eos. Monocytes and the granulocytes are phagocytes, which means they phagocytize things, and that just means that they eat them. They engulf bacteria to destroy it. Lymphocytes produce antibodies, and the antibodies can fight off bacteria or viruses for us. The granulocytes are called granulocytes because they have granules. You can't really see the granules too good in these pictures, but they're found in the cytoplasm. Granulocytes have more than one nucleus. See, the neutrophil has these little segmented parts, the EO has them, and the baso. Monocytes and lymphocytes have one nucleus. This is all one nucleus, and this is all one nucleus. So the neutrophil has pale, fine granules. The EO has orange to red granules, and the baso has dark blue to black or even purple looking granules. The thrombocytes are also called platelets, and they're involved in clotting. The platelets are not actual cells, but they come from one big cell, and they're actually little pieces of a cell. They don't have a nucleus, they don't have a cell wall, and platelets are only viable for about 3 days. Your notes say a few days, but it's about 3 days. And what they do is they interact with the clotting proteins to help form a clot whenever there is bleeding or a reason to form a clot. Okay, the CBC is a test that we do for all of these cells, and CBC stands for complete blood count. On the blood count, this is what if we were to analyze a patient's sample, this is an example of the results from the instrument. This is the white blood cell count, which is 8.8, and notice that it's really 10 to the third. So it's 8,800, but we get, this is the result that we release, 8.8. Down here, this is the red cell count, 4.24, and notice that's 10 to the 6. The hemoglobin is this, it's 12.4. The hematocrit is 37.3. You don't need to worry about these. Under here, this is the platelets. The platelet count is 344, and notice, notice this is times 10 to the third, so this is really 344,000. Also up here, this NE stands for neutrophil, which is one of the granulocytes. The LY is for lymphocytes. The monocytes is MO. The EO is eosinophils, and the basophil is BA. And this does a percentage. So this is the total white count. And then what it does is it counts at least 100 white cells, and and out of that 100 white cells, 65.7 are neutrophils, 25.7 are lymphocytes, 7.9 are monos, 0.5 are eos, and 0.2 are basos. And that's called a differential. Okay, so it gives us red cell, white cell, and platelet counts. Hemoglobin is the amount of the oxygen carrying capacity of the red cells. The hematocrit is the percentage of packed red blood cells. So there's a picture in your notes if you got your notes in front of you. And this is what, if you were to take a little tube of your blood and you pack the red cells down all the way to the bottom, this is what percentage of the total is your red cells, is basically what that is. And then the white cell differential is the percentage of the different types of white cells. The instrument does this automated differential of the white cells, but we can also do a diff, and that's what we say for short is a diff. We can make a blood smear and stain it with a polychromatic stain called the Wright stain, and we can do a differential ourselves. And when we do that, we count 100 cells and we report out how many neutrophils, how many lymphocytes, how many basos that we get. And the reason that we do that is because people who have abnormal things going on, like leukemia or other types of things going on with their white cells, the instrument can't count the really abnormal patients. So we have to do it under the microscope as a backup. The CBC is done on EDTA, which is a purple top, and it's done on whole blood. So what that means is we don't centrifuge it. We mix it and we analyze it like that. The PT and PTT are clotting time tests. The PT stands for prothrombin time, and the PTT stands for partial thromboplastin time. These are done in hematology and coag. So usually hematology and coag are grouped together in most labs. So the CBCs are done in hematology and PTTs are done in coag. So they're kind of done in the same department. So this tests the availability and the function of clotted proteins, the PT and the PTT do. So it tests for the clotting proteins and their ability to make a clot, because you may have the clotting proteins, but they may not work properly. So it tests to make sure that they're not only there, but can they make a clot? Are they working properly? And who has this done are people who have bleeding and clotting problems, and also people who are on blood thinner. They use the PT to monitor patients who are on blood thinner at home. And this is done on citrated blood. So this is a blue top in a sodium citrate tube, and these are centrifuged down, and we use the plasma to do the test. All right, body fluids. We do cell counts on the body fluids, like I said before. We do the red cells and white cells. We not only do spinal fluid, but we do the serous fluids and synovial fluids. So we do pleural, we do pericardial, and we do peritoneal, which is in the abdominal cavity. So the spinal fluid is the most important. It's always considered stat. That means we do it right away above all other tests. So when a patient has a spinal fluid test, they're usually looking for either meningitis or if they've had a cerebral hemorrhage. Those are the two main reasons why we would collect spinal fluid. So the doctor collects spinal fluid by lumbar puncture. So he actually goes into the lumbar column and draws off fluid. He only draws off a small amount because there's not a lot of fluid in the first place. So only about 2 milliliters is collected, and it's collected in different tubes, and the different tubes go to different departments, so we can do them at the same time. So hematology will have its own tube, and this is where we do the red cells and the white cell counts. Chemistry does a protein level and a glucose level. When a patient has meningitis and it's due to bacteria, the glucose level will be really, really low. In micro, they get their own tube. We do a culture, and if anything grows, then we do a antibiotic sensitivity. So your notes say CNS, and that stands for culture and sensitivity. The third tube that's collected is usually sent to micro, so if there's any contamination from normal flora on the patient's skin, hopefully it will be gone by the third tube. The lumbar puncture is performed on patients that have symptoms of meningitis or hemorrhage, and it's done between L3 and L4, which are the vertebrae of the lumbar. Patients who have meningitis, they're not always children, but usually this is the ones that more commonly have it. The number one sign is a stiff neck, but also they can have really high fever and headache. Spinal fluid usually looks like water, so it's clear and colorless. If the patient has a cloudy white specimen, that usually indicates WBCs, and WBCs means they have an infection, which could be viral or bacterial. A cloudy red specimen indicates that there's red cells there, and that can either be due to a traumatic tap or a hemorrhage. A hemorrhage would mean that they actually have a brain bleed. A traumatic tap is not something that is wrong with the patient. A traumatic tap is when they collected the spinal fluid, they accidentally hit a artery or a vein, and there is blood in there, which is not pathologic. There's nothing going on with the patient. And that's it for this module.